Assalamu alaikum. Good day for you all. Here we are together again. I'm Dr. Marwan for TBYP, or Tips to Boost Your Knowledge in Pharmacology for medical students and uh, other students of medical background. This is step number 7 about the design of the blood-brain barrier and its effects on drugs and chemicals. So the objective of this tip is to briefly compare between the liver and the brain and the fetomaternal circulation, capillary tissue barrier design. Also, to compare between the liver and the brain capillaries in terms of P-glycoprotein function and the glucose transport mechanism. So, to start with the blood-brain barrier, or to start with the capillary hepatocytes barrier here, we can see that the cell membrane at the capillaries, and this is the lumen inside the capillaries, where many things can be transported from the intracapillary side into the hepatocytes because of the presence of large and wide gaps between the capillary cell membrane. Uh, while in the same capillary at the brain area, where we call it the blood-brain barrier, we can find that these junctions becoming very tight and very narrow, which blocks the transport of many water-soluble molecules and drugs, allowing only the lipid-soluble drugs to penetrate through the lipid bilayer cell membrane. So for the charged drugs or the water-soluble drugs, they can only pass to the other side of the brain, from the brain capillary to the brain tissue, by means of carrier-mediated transport mechanism. So this is a protective mechanism to the brain, while it's an open mechanism for drug and chemicals to enter into the liver. Here, this is what you call another illustration of showing you how the liver allows large molecules of water-soluble drugs to go through the very wide and very large gaps that exist between the cells of the capillary wall, uh, and also even the lipid-soluble drugs and molecules can penetrate also through the lipid cell membrane. While in the blood-brain barrier, as we've just mentioned, the presence of the tight junctions will block the water-soluble molecules, and therefore they will not be able to enter. It's a protective mechanism. The only way for the water-soluble molecules to enter is through carrier-mediated transport, as we have just mentioned. In the fetal-maternal circulation, only the lipid-soluble can pass through from the maternal to the fetal circulation, while polar drugs, water-soluble drugs, are blocked because of the presence also of the tight junctions. So the other mechanism that we want to discuss is the P-glycoprotein, which is a pump that is available, a type of glycoprotein, which is a pump that's available almost on the cell membrane of almost all cells of the body, especially the capillaries that are present in the brain, the liver, and the fetal-maternal circulation. So to compare between the three, we see that the P-glycoprotein at the liver, it transports and pumps a variety of drugs into the bile, so away from the circulation, because this suits the function of the liver as an organ of detoxification. However, in the blood-brain barrier, the P-glycoprotein pumps the drugs out of the brain back to the circulation. So this type of P-glycoprotein at the brain area functions to pumps the drugs and chemicals away from the brain, not towards the brain. For the placental P-glycoprotein, it pumps the different kinds of drugs out of the placental cells back to the maternal circulation as a protective mechanism for the growing fetus. So, in brief, the liver being the major organ of detoxification and drug metabolism, we find it that it is allowing uh, uh, a large number of drugs through the large capillary gaps that are available to have water-soluble drugs penetrate and enter the hepatocyte for metabolism and detoxification, while the brain does not allow uh, those water-soluble in large molecules because of the presence of the tight junctions. Second is the presence of P-Glycoprotein with different functions. The cell membrane of the blood-brain barrier, for example, which pumps back the drug back to the circulation. Whatever drug that passes through carrier-mediated or carrier-mediated mechanism, uh, or even the lipid-soluble drugs, they can be pumped out back again as a protective mechanism to the brain. While at the hepatocytes, you can find that the same P-glycoprotein has a different function, which to pumps the drug into the bile away from the circulation. And of course, at birth, the neonates are considered to be more vulnerable for having CNS toxicity because the blood-brain barrier is not well or not fully developed. Uh, Now, talking about the third uh, point for the tip today about glucose transport across uh, the capillaries into the CNS and across also the other structures of the body, like the skeletal muscles, we find that glucose being the major source of energy for the brain metabolism and even for action potential, to provide energy for action potential, as we can see in this illustration. Glucose, we find it passes in an insulin independent mechanism through GLUT1 transporter. GLUT1 is glucose transporter number one, and it is available in the blood-brain barrier, and it does not depend on insulin, unlike GLUT4, which is a transporter that is responsible for the transport of or the movement of glucose into the skeletal muscles, the adipose tissue, and the heart. Those transporters, GLUT4, at the skeletal muscle are insulin dependent, means in the presence of insulin, then only the glucose will be able to be transported from the circulation into the skeletal muscles, adipose tissue on the heart. So, and despite the presence of these tight junctions that we have explained earlier, we find that glucose enters the brain in an insulin independent manner through GLUT1 and GLUT3. Yes, and uh, this is considered as a life-saving design at the blood-brain barrier for people with diabetes. And I want to hear from you in the forum as a point of discussion. Why is it considered as life-saving to have glucose transporter 1 and glucose transporter 3 as insulin independent, as a life-saving design for the blood-brain barrier, especially in patients with diabetes? So with that, we have to know that the design is not haphazard, is not random, of the capillary tissue binding barrier, and this will give us a hint about how intelligent is the design of our body, including the transport of material and substances into the brain and into the liver. How does it differ? And this is a message from the intelligent designer about his intelligent design. Thank you so much, and see you again with another tip to boost your knowledge in pharmacology. Assalamu alaikum wa rahmatullah.